Liquid cell electron microscopy is a powerful technique to investigate nanofeatures in situ in liquid media at high resolution and provides unique, real-time insights into dynamic processes at the nanoscale.
The graphene-supported microwell liquid cell combines the advantages of both graphene and silicon technology-based cell architectures. It allows for correlation with analytical methods such as EDX spectroscopy on-site.
The technique enables us to directly follow materials processes in liquids while they happen. This in situ observation is in the very core of our research training group, in situ microscopy with electrons, X-rays and scanning probes, funded by the German Research Foundation. Demonstrating the procedure will be diploma chemist Robert Branscheid, a staff member from my laboratory.
To begin, transfer the graphene onto TEM grids, first wetting the tissue supporting the 6 to 8 layers of CVD graphene on PMMA. Take care that the water is not directly applied to the PMMA membrane. Fully immerse the PMMA-coated graphene in a petri dish filled with DI water, and then use filter paper to scoop up the graphene layer. Take care that the graphene side of the graphene PMMA stack stays on top during the whole procedure. Cut the graphene layer into pieces that are large enough to cover all of the fabricated wells. Then, re-immerse the cut pieces into the petri dish. Next, using a pair of anti-capillary tweezers, pick up a TEM grid coated with a support layer of holy carbon. Carefully dive the grid into the water and catch the graphene floating on the surface. Let the sheets